Hello guys and welcome back to Excel Academy, lecture 14. And in the last lecture we have discussed about the different type of toxins, right? So now we are going to talk about the inophores. What are inophores? What are the different types of inophores? Now, so again, if you see, there are two types of inophores. If you have understood how the protein or any molecule is getting transported, so we have got the carrier inophore, like they carry the ions, and without them coming in contact to the hydrophobic area, they just flip and turn towards the inside. Yeah, out, yeah, inside ho gaya. Or we can have channel forming inophore. So again, this does not have to come in direct contact of this hydrophobic region. Okay, so there are two types of there, and that is what we're going to discuss now. Okay, so they are hydrophobic molecules which is produced by microorganisms that facilitate the ion transport across the lipid bilayer of the cell membrane. So two types are there: channel formers and the carrier compound, just now as we saw in the figure. So channel formers, these inophores, they make a hydrophilic pore in the lipid bilayer, which will allow the ion transport by avoiding the contact with the lipid bilayer. Okay, so जैसे भी हमने देखा, you have got this plasma membrane is there, and it forms like a channel. So directly without coming in contact to the lipid bilayer, it is helping in the transport of the ions from outside to inside or vice versa. Now, carrier compounds. These are the mobile ion carriers which facilitate the transport of ions through the lipid membranes, again by masking their charge. So let us say that Na plus है, Na plus है, तो अभी वो उसको directly contact में नहीं ला सकते hydrophobic area के, right? So there are these proteins, they will make interaction with Na plus and then they will flip, and Na plus will be taken to this side without that is like masking their charge and without coming in contact to the hydrophobic area. Okay, simple. So inophore simply work as an antibiotic, disrupts the ion gradient which is necessary for normal cell functioning. So अब हम जो discuss कर रहे हैं, in short we are discussing how antibiotics are actually acting or killing the bacteria. Okay, so it's a potent act of defense among the microbes with their competitors. So usually क्या होता है, जैसे in the last lecture I was explaining that there are certain gram-negative bacteria which produce certain kinds of toxin or antibiotics. Okay, now this will be in response to their competitors, or for example against the gram-positive bacteria. Okay, so what we as a human, for the welfare of our society, what we are going to do is we make use of this कि हम ग्राम नेगेटिव के एंटीबायोटिक्स को हम ग्राम पॉजिटिव के लिए यूज करेंगे, और जो ग्राम पॉजिटिव रिलीज करता है इन रेस्पॉन्स टू द ग्राम नेगेटिव और अदर competitors, we are going to use that in order to treat the infections which is happening to the humans. Okay, so let's see a few examples. Now, conductive carriers, for example, Valinomycin, which is for K plus, hemisodium Na plus, FCCP, triple CP, H plus. Electron neutral exchangers are there, nigericin, K plus, H plus, channels, gramicidin and nistan. Gramicidin याद है कहाँ पे पढ़ा था हमने? In the last previous lectures we have discussed about the gramicidin, right? It comes under the category of small linear peptide toxin, right? In that we have discussed. Now let's talk about these inophores. एक एक example हमने लिया है. For example, calcimycin, नाम से ही पता चल रहा है that it is for the calcium. Okay, it is produced during fermentation by streptomyces. And one more thing which you have to understand here is that streptomyces species से ही maximum antibiotics are being produced. Okay, now the concept, the evolution, like क्यों वो इतने antibiotics produce करता है? Who can tell me? Who have got the idea? Akshaya, would you like to try? Like why the different species of streptomyces or the other organisms are keep on producing antibiotics? In short, why organism is producing antibiotics? Then we can discuss about the evolution. Shruti, do you want to try?
It might give them a competitive advantage against the other microbes.
Correct, correct. So it's like, see, in a environment where all species are existing, okay, the food source or the resources, other resources are limited. Correct? Now, once you start producing antibiotics or toxins, which is going to kill your competitors, what exactly you're doing? You're making sure that in future you have got the resources available for you for your progenies. And that's why they keep on changing or keep on improving the antibiotics. ऐसा नहीं कि सिर्फ streptomyces करता है, bacillus भी करता है. So it is like the race कि ये एक अगर improve कर रहा है तो मैं भी improve करूंगा. So this is how, depending on the availability of the resource, they keep on changing the antibiotics. And we as a human, what we are going to do, we make sure that these antibiotics we take, because अगर कल को अगर streptomyces से अगर infection है या staphylococcus से है या bacillus infection है, so I'm going to make sure that the enmity between these organisms, they are going to produce the antibiotics. So I'll make sure that I'll take these antibiotics and treat, because at the end of the day, I don't want any of these three species infecting me. है ना? तो मैं इस चीज का एडवांटेज उठा के उसको मैं अपने फायदे के लिए यूज करेंगे. And that's why now also many research is going on. Till now, from streptomyces, so many so many antibiotics are released. सुना भी होगा, एमॉक्सिलिन हो गया, स्ट्रेप्टोमाइसिन हो गया, एरिथ्रोमाइसिन हो गया. So all these are nothing but the antibiotics released from these bacteria only. Okay, now coming about the evolution, it says that only, like survival of the fittest. जो जितना अच्छा एंटीबायोटिक बना लेगा, उतना अच्छा वो competency रखेगा, and it is able to kill their enemy, so that the food resources available in the nature will be taken by them. Correct? So that's how they keep on increasing the versions of the antibiotics. अब इसकी वजह से क्या होगा? They also get resistant. जब आप कुछ improve कर रहे हो तो obviously the organisms are also becoming resistant. ऐसा तो है नहीं कि जो 100 साल पहले एक जो दवाई चली थी, now also it is going to be beneficial. No. Why? Because over these 100 years or this 10 decades, the organisms have become resistant to those antibiotics. So what is the need now? The arm race. इसका मतलब क्या? अगर आपका organism resistant हो रहा है, so I also need antibiotics which is going to kill this resistant organism. It means this antibiotic should be also advanced. अब क्या होता है in lab and all, where these pharmaceutical companies are there, what they do is that, for example, this is a drug existing. Okay, हमें मालूम है कि let us say that it is killing some certain amount of bacteria. Okay, I just want to make it a more advanced. So what I can do, I can change these functional groups. Or I'll just see like key functional group change करने से whether I am making it more potent or I am destroying its activity. तो यहाँ पे the role of organic chemistry आता है. So these people, they know like what kind of reaction should be carried out so that they can change the functional groups over here. Then we have got the other people, for example, as a computational people जो होते हैं, they will check in the computer by running the simulation studies whether this particular drug is able to kill the bacteria or not. Killing the bacteria means whether this drug is able to bind to the receptor present on the bacteria. ये computational वाले काम करेंगे. And if this association is tight, then of course it says that this is going to kill the bacteria. अब हमारे जैसे माइक्रोबायोलॉजिस्ट जो बायोटेक्नोलॉजिस्ट होते हैं वो क्या करेंगे? अब हम उसको बैक्टीरिया में लैब में चेक करके देखेंगे इन विट्रो. Okay, and then we talk about yes whether there should be this drug should be released in the market or not. तो ये बहुत लंबा प्रोसेस होता है. ठीक है, about the drug coming into the market. Okay, coming back to the topic. So it is produced during the fermentation by the streptomyces. They act as inophores for their divalent cations. जहाँ पे भी आपका 2+ रहेगा. Example, calcium 2+, Mn2+, Mg2+, strontium 2+, and barium 2+. Okay. So what it does is that calcimycin, it is uncoupling the mitochondrial ATP synthesis activity. So uncoupling the mitochondrial ATP synthesis means what? Who can tell? Tanvi, would you like to try? Uncoupling मतलब क्या होता है यहाँ पे? If you remember, we have discussed about the P pump and F pump, V pump. तो उसमें हमने ATP synthase के बारे में पढ़ा, right? Now, this is a hint which I am giving you. Now, tell me what this uncoupling means. Okay, Akshaya, would you like to try again? No. Shruti?
Does it prevent the subunits from working together, or does it cause disengagement of the ATP synthesis complex from the membrane, or
It is the
something like that?
first one. So, जो हमने F0, F1 particle याद किया था, पढ़ा था, right? So, one is responsible for, taking the H plus out and making the proton gradient. And then when it is passing, so what happens? There is a one motor, right? Gamma motor, which rotates and converts the proton gradient into mechanical energy. Mechanical energy फिर क्या करता है? ADP plus PI, ATP देता है, right? So that is being broken. So the functioning of F0, F1 particle, this coordination is being broken. Okay, good. At least you tried. Okay. Now, binding of calcimycin, it activates NF-kappa B dependent pathway in macrophages. What is NF-kappa B over here? It is your transcription factor. Okay. Now, NF-kappa B dependent pathway regulates interleukin stool production upon calcimycin treatment. So these secreted interleukins modulates autophagic pathway in an autocrine fashion. Autocrine matlab self-signaling. Autocrine means self-signaling. Correct? अगर कोई पर्टिकुलर सेल है, ये जो भी रिलीज करेगा वो उसी रिसेप्टर को बाइंड करेगा टू द सेल्फ रिसेप्टर. With its receptor IL-2 binding with its receptor phosphorylates JAK2. It induces autophagy, inhibits intracellular microbacterial survival. So now calcimycin treatment at the end, it is talking about the mycobacteria. Now why mycobacteria? Because see, if you know mycobacteria जो आपका TB cause करता है, okay, these mycobacteria are engulfed by the macrophages, okay, by the वही जो autophagy हम बोलते हैं, ठीक है. Now inside the macrophages they try to kill the bacteria. Now वो कैसे होगा? This is the way, okay, like at the end what we want is the mycobacteria should be dead. Now this is a pathway which is being talked about. Let's discuss the pathway here. Slowly we'll go. Okay. Okay. So now we have got the calcimycin, which goes and binds with receptor. Okay. This is one part. We also see Mycobacterium tuberculosis over here. So what is happening? It gets inside. Okay. इसको हमने खा लिया. Induction of the autophagy. So it is over here. And now what is happening? It is now getting engulfed, and what will happen? Finally, जो आपने खाया है, it goes and binds to the lysosome. If you know, lysosome में हमेशा आपका degradation होता है. Whatever you engulf inside, it goes and binds to the lysosome. If it is autophagy, then it becomes an autophagosome. Is this much clear? Okay, this is one part. Here what is happening, when calcimycin treatment is given, it binds to its receptor and activates your NF-kappa B pathway. Okay, अब यहाँ पे क्या हो रहा है? ध्यान दो. It's actually multiple signaling together happening, that's why. So just focus over here. So when calcimycin is binding to its receptor, the NF-kappa B pathway is getting activated. Now, in the cytoplasm, when there is no signaling, NF-kappa B is bound to the I-kappa B. I-kappa B is the inhibitor kappa B. Okay. अब क्या होता है? When this is getting activated, it means there are certain kinases which goes and phosphorylates them. Upon phosphorylation, I-kappa B loses its affinity for NF-kappa B. ठीक है, I kappa B goes for the degradation. Now you have got NF kappa B activated. As I mentioned before, NF kappa B is a transcription factor. So this will be recruited inside the nucleus where it goes binds to the gene and you give the interleukins production. Okay. Now interleukin is getting produced. What is happening? They go and get released. Kya? This is your autocrine signaling now. Interleukins जो प्रोड्यूस हुए हैं वो आपके बाहर जाके रिलीज होंगे. Since it is autocrine, this interleukin 12 will go and bind to its receptor, self receptor. Okay, इतना clear हुआ? See, same thing. Interleukins comes and binds to the receptor. Interleukins stay which comes and binds to its receptor. Now, binding of this receptor of interleukin, okay, it is going to activate your JAK kinase pathway. This is your JAK kinase pathway. Now, JAK kinase pathway when it gets activated, it is going to activate or the induction of the autophagy, where the mycobacteria which is got engulfed by the macrophage will go for the autophagosome, formation of the autophagosome. So then it gets fused with the lysosome, resulting in the formation of autolysosome. Now, abhi kya ho raha hai? In the autolysosome, you have got certain enzymes which is going to kill the Mycobacteria tuberculosis. Guys, clear? थोड़ा सा बड़ा था, but I hope you got the point. I can repeat again if it is required. Shruti, what is your view? Should I repeat once again because
I'm
so
clear.
many? It's clear? Yeah.
For me, it is.
Okay. What about rest? Akshay, Tanvi?
Oh, ma'am, can you repeat?
Okay, I'll repeat. No
In
problem.
short.
Okay. So, see, we'll start from the calcimycin treatment. Okay. So when calcimycin treatment is given, it goes and binds with receptor. Okay. Okay. First, you need to understand. Huh?
Now, can I try saying the pathway?
Huh? Okay. Okay. Yeah. Sure. Good. But before you start, I just want to make sure that this calcimycin treatment is to kill the mycobacteria. Okay. Now, how it is doing by the induction of this IL-12. Okay. Yeah. Yes, Shruti.
Okay. So there's this thing called calcimycin. It goes and binds to its receptor. And when the receptor is there. So usually without the calcimycin, there's this NF kappa B, which is always bound to I kappa B. And when the receptor is activated, that causes I kappa B to be phosphorylated and goes through the ubiquitin pathway, which frees up the NF kappa B to go and bind to the go through the nucleus and bind to the gene that causes the transcription of interleukin 12. And then interleukin 12 goes outside the cell, but then since it's self-signaling, the same cell has the receptor for interleukin 12 as well. So that receptor is bound, and then it activates the J kinase pathway, and that causes activation of for autolysosomes to happen, as in the mycobacterium to undergo to get autolysosome. Like they form this pathway where autolysosomes happen, and then lysosomes enzymatically kills the bacterium.
Correct. Absolutely correct. So this is how macrophages is killing the bacteria. Okay. I hope it's clear, Tanvi.
Yes, ma'am.
Okay, cool. Nice. Okay, now something else. So वो हो गया आपका. Now ionomycin. वो था आपका calcimycin. This is ionomycin. So it's a membrane permeable calcium ionophore which is produced by the bacterium Streptomyces conglobacterus. Okay. Now this ionophore, it facilitates the transfer of calcium ions into and out of the cells. It can be used to increase the intracellular calcium levels by triggering cell death through apoptosis and autophagy. Okay. Now अभी ये भी देखो कैसे. So जो अभी हमने डिस्कस किया था वो क्या था आपका रिसेप्टर, चैनल बेस्ड रिसेप्टर था, राइट? Calcimycin goes and binds and activates. Now here it is your, can you see small molecules आते हैं. So there these are your carrier molecules. जो हमने दो तरह के देखे थे, एक चैनल होता है, एक आपका कैरियर होता है, right? So here this one is your carrier ones. So carrier ones, they take the calcium and just flip towards the inside. So calcium का जो charge है वो आपका hydrophobic area में नहीं आएगा. Okay, now look it over here in this case. So what is happening? The calcium is there. Okay, inomycin लिया, calcium क्या हुआ, RBC में बहुत सारे आ गए. Okay, now this, when you have got more amount of calcium, it results in the disbalance. K plus Cl minus water dehydration में result करेगा. So it is killing the cell now. ये क्या हो गया, vanadate. If you remember what is vanadate, क्या था? Did you remember what is vanadate? It is actually an analog of dash. सुना हुआ लग रहा है? If not remembering. Okay, hint is also given there. PMCA क्या था? एक सरका था और एक PMCA था. No one? प्लाज्मा मेम्ब्रेन कैल्शियम और एक सरका, क्या था आपका सरका? सार्कोप्लास्मिक रेटिकुलम कैल्शियम. याद आया कुछ? मसल्स में कैल्शियम चैनल्स. Okay, so those are your calcium ATPases. Vanadate क्या था? Analog of phosphate. If you remember हमने क्या दिखाया था? इससे phosphate का आपका structure रहता है. Similarly, P के जगह पे V रहता है. So because of the structure similarity, it was able to inhibit the channel. ठीक है? That's okay. Once you go through again, you will get the answers then. Now coming to the sodium ionophores. So अभी तक हमने कौन से देखे थे? Calcium. Now this is your sodium. Example is monosin A. So it is a polyether antibiotic which is again isolated from the streptomyces cinnamonesis. अब देखो cinnamonesis, इसलिए इसका नाम भी monosin A रखा है. ठीक है? Now this is used in the case of the ruminant animal feeds. For example, your cow, buffalo. ठीक है? Now why it is used in the beef and dairy industries? Because to prevent the coccidiosis, which increases the production of propionic acid and prevent the bloat. यानी कि क्या? See, when the feed is not good, and just imagine that there certain bacteria are there or certain उनको क्या कहते हैं, protozoas are there. They get inside of the ruminant animals. ठीक है? And then they start infecting the animals. So क्या होगा? फिर gas बनना शुरू हो जाता है. That is like in the form of the propionic acid. And this the animal starts bloating, जिसकी वजह से दूध अच्छा नहीं आएगा, जिसकी वजह से काऊ डंग अच्छा नहीं आएगा. And from the dairy industries in which we are interested at the end of the day, in the milk and the cow dung, right? So that problem will be there. So to prevent all this, they add monosin. Okay. Now it transports monovalent cations such as lithium, Na plus, K plus, etc., etc. It is able to transport these cations across the lipid membranes of the cell in an electroneutral fashion. यानी कि क्या? There is no charge का. It's like playing important role as an Na plus H plus antipoder. यानी कि क्या? एक plus गया है तो दूसरा भी plus जाएगा. So the charge is getting balanced. Okay. This is the example. Gut lumen है और यहाँ पे आपका Na plus है. So inside outside. So inside the parasite it is going so that the parasite is able to we are able to kill the parasite. Mainly it is the protozoas and the other parasites which is present in the gut lumen. Okay. Now coming to the valinomycin. Now valinomycin is important because ये exam में आ चुका है. This is important one. तो अभी तक हमने जो भी डिस्कस किया, कैल्सीमाइसिन एक बार पूछा एग्जाम में और वैलिनोमाइसिन पूछा है. It doesn't mean that कि सिर्फ यही आएगा. So we should be prepared for the rest. So it is a natural neutral inophore. It again it is obtained from the streptomycin, sumensis and streptomyces fulvicimus. Okay, it is a dodecadepsy peptide, highly selective for K plus. I think this means 12. Dodecadepsy. Valinomycin was recently reported to be the most potent agent against the SARS-CoV. Now see the structure. What is it actually? It is the three size, yeah, na? So, 1, 2, 3, 4. So 4 3s are 12. Yes, it is your actually 12 peptide. Correct. So you can see it's valine, dihydroxy isovaleric acid, valine, and then lactic acid. Okay. So this is what you have to remember, and it is the thrice of it. Valine हो गया, valeric acid हो गया. Now please do remember that this is L-valine and here it is D-valine. ठीक है, ये भी आपको ध्यान रखना है. And then the lactic acid. So this is your valinomycin. Again, it's a peptide structure only. This is again same. You can see over here, right? Valine, L-valine, isovaleric acid, then D-valine, lactic acid. Again, valine, isovaleric acid, D-valine and lactic acid. तो ये इसी की तरह से एक आपको ये बनता रहता है. ठीक है? Clear, guys? This one. Okay, this just the same thing. Representation is different. Okay. Yeah. Any questions from this Inophores? Okay, so takeaway message of this topic is Inophores क्या है? Do not get confused Inophores with the ion channels which we have studied before. ठीक है? आपको Inophores क्या है? Ion channels हो गए, voltage gated हो गए, and then ligand gated हो गए. ठीक है? हर एक में we have got the example of Na plus and K plus. So कल को अगर कोई नाम आ रहा है, so you shouldn't get confused that these are the Na plus channels. No, Inophores अलग है, channels अलग है, voltage gated अलग है, ligand gated अलग है. Okay, so please do understand the difference in these. और आज जो हमने पढ़ा है उसमें से calcimycin, then valinomycin is important. इसका mechanism, how it is helping in killing the mycobacteria, valinomycin का structure. Okay, anything else, guys? This is the takeaway message. Like any questions from your side? So I'll be stopping here then. And tomorrow, 11 o'clock, there is the last topic about the what is exocytosis, endocytosis, that we will complete. With that, membrane salute is getting over. And one more suggestions, there are certain interview-based questions on these membrane. I'm sharing in the group also, but please keep following so that you don't have to prepare separately for the interviews. Right now, interview sessions are going on for the different students. Okay, so that's why I started preparing again for them. So you guys also keep watching because आपको फिर अलग से interview के लिए prepare नहीं करना पड़ेगा. Okay, anything else? Akshaya, Shruti, Tanvi. Hey Tanvi, I didn't ask you. Your exams are over, right? How was it?
Yeah, ma'am, they were good.
Good, good.
I have practical on 24th.
Okay.
Hmm.
Yeah, so any doubts from this topic? Then we'll be starting new topic from tomorrow onwards. Nucleus से चालू करेंगे after endoexocytosis and all. We'll start with that. Evening, I'm not sure. I'll let you know, but definitely 11 o'clock we have class. Okay. Anything else? And Shruti, Akshay, did you guys applied? Akshay, I know. Shruti, what about you? Did you apply? Any response?
Response not yet. I mailed 5 people.
Okay. Keep applying then because 5 is not sufficient. Okay.
Okay.
Okay. Okay then guys, I'll be stopping here now. Okay. And I'll try to get the questions also so that we can go through like what kind of questions are coming from yesterday's topic and today's topic. Okay. Yeah. Chalo, bye bye.
Bye-bye, ma'am.
Good night.
Thank you, ma'am.
Thank you.
Thank you, ma'am. Good night.